Good afternoon. Welcome to our Calleva Seminar for 1st of May, which of course is an important day at Magdalen. So I don't know if any of you were up for the singing on the tower. I certainly had to be. But I hope that you've had a good day, and we're all here to enjoy Rachel Wheatley's seminar. So Rachel was a fellow by examination or junior research fellow here for a few years and has just taken up a faculty position at Queen's University in Belfast. And Rachel received one of our Calleva Research Project Awards. I was going to say something very briefly about the Calleva Centre. So the Calleva Centre for Evolution and Human Science was founded about the year 2009 here at Magdalen through the generous benefaction of Stephen and Caroline Butt. And since then it's grown both in terms of its resources and in terms of the kinds of projects and the range of types of projects and types of research it can do. At the moment we have a particular focus on risks to humanity, and there are a whole range of projects that have been funded in that space, whether it's to do with crop science, and whether it's to do with climate econometrics, whether, as in this case, it's to do with looking at antibiotic resistance, but also whether it's to do with how despots use language, or would-be despots use language, or how better vaccines can be developed. That kind of area too is something that we're interested in, or the history of pandemics. So there's a whole range of different aspects of science which covers risks to humanity. And Rachel's project was something we were really glad to fund because it provided her, as a relatively junior member of the college, with a research assistant and an intern, which was a great opportunity for Rachel, a great opportunity for the student, and enabled some work to be pushed forward that you'd been doing and developing as a distinctive aspect of your research interest during your junior research fellowship. So it's lovely to welcome Rachel back now that she's started her faculty position in Belfast to give our seminar for today. And at the end, please do ask questions, and there'll be a mic, I think, to carry around to amplify any questions. Welcome also to those who are joining us online, because this is simultaneously being webcast as well as being performed in person here. And do please stay at the end for a drink and to ask Rachel any follow-up questions you want to ask her afterwards. So over to Rachel, and thank you so much for coming back over from Belfast for this seminar today. Thank you.
Thanks very much for the introduction. It's really nice to be back here in Magdalen. So my name is Rachel, and I'm going to talk to you today about some of my research at the interface of ecology and evolution, and trying to understand antibiotic resistance in bacterial pathogens. So my talk today is going to be split into 3 parts. So I'm going to start off with quite a general introduction to antibiotics, the kind of history of antibiotic discovery, which excitingly includes some stuff here in Magdalen. And then a brief overview of what are antibiotics, how do they work, and what do we know about how resistance evolves. I'm then going to give 2 examples from my research. So thinking about what can we learn about resistance evolution from studying this process in real patients, and then what can we learn from the kind of differences and what's going on in patients, and then take this into the lab to dissect further in a controlled environment. And I'm going to wrap things up with talking about some perspective across the field. So I've introduced the problem of antibiotic resistance, but overall what's going on in this space with regards to how can we combat it moving forwards. So bacterial infections have been a real major killer throughout history. So a really well-known example is the bubonic plague, or Black Death, which kind of ravaged throughout Europe and the rest of the world during the Middle Ages. And it's estimated to have killed around 1/3 of the total world population during this spread in the 1300s. And we now know the kind of causative pathogen implicated behind the bubonic plague is this bacterial pathogen called Yersinia pestis. Another example, kind of moving forward, that a lot of people will know about is tuberculosis. So tuberculosis has been a really big problem for, well, hundreds of years. We've got the kind of bacterial pathogen here, Mycobacterium tuberculosis. And what I think is quite interesting is, so with tuberculosis, a standard treatment before our use of antibiotics was fresh air. So this picture here is a photograph taken in London in, I think it was 1936, which is showing some patients with tuberculosis getting this fresh air treatment. And so I think it goes without saying that antibiotics are pretty incredible, right? They've saved millions and millions and millions of lives. And if we look at this graph here, so this is showing estimated deaths from infectious diseases per 100,000 people. And so antibiotics are estimated to have helped reduce global deaths from infectious diseases by over 70% since the 1940s. So over here in 1942, we've got this first use of the antibiotic penicillin. And penicillin has a pretty cool story behind it, which maybe some of you will have heard about. So it was actually discovered accidentally in 1928. And this is Alexander Fleming, who was working in London. And the story is, so Fleming was growing the bacterial pathogen, Staphylococcus aureus, on these agar plates. So these are plates that you can grow bacteria on that have lots of the kind of helpful nutrients they need to grow. And he was growing these little white dots here, are these Staphylococcus colonies, and he was growing up this bacteria on his plates. He went away on holiday and he came back and noticed that his plates had started to grow some mold or fungi. And I think the funny or nice thing here is, if you're also a microbiologist, you'll see that in 100 years, this problem has not gone away from the lab. It is still quite easy to get your plates contaminated with fungi. But the interesting thing that he noticed is, so this is the fungi or penicillium colony. And he noticed there was a zone of inhibition around this colony where the bacteria weren't really growing so well anymore. And we can see this perhaps a little bit more clearly in this kind of modern replica of the experiment, where what we're having here is, so the Staphylococcus bacteria is this white smudge you can see on the plate, and so it's been streaked out in this zigzag pattern. And then a plop of the kind of penicillium colony has been put there, and you can really clearly see this zone around it where the Staphylococcus colonies aren't growing. So this made him think, okay, this looks like this fungi, this penicillium, is producing something that's killing or inhibiting the growth of these pathogenic bacteria, which is, of course, sounds really important and great, right? And so Alexander Fleming and the team worked on this for quite a few years, trying to work out, what is this compound, what can we do with it? But it wasn't till 1941 that people were actually able to purify this and use it in a first effective injection. And really instrumental in this process were Howard Florey and Ernst Chain, who were really important for working out how to purify it and how to use it in injection. And I think there would be something amiss if I gave a talk at Magdalen about penicillin and antibiotics and I didn't talk about Howard Florey. So Howard Florey was an Australian microbiologist who actually came to Magdalen as a student, as a Rhodes Scholar for his PhD. And he was really important in this process in working out how to purify penicillin. And if you go around Oxford, you can see these blue plaques that we've got stuck up everywhere. And you can see two really neat ones relating to penicillin. So we've got on the Dunn School, you can see if you can go and find this. So it says, in this building, Howard Florey, Ernst Chain, Norman Wheatley and colleagues first isolated and purified penicillin. And then over here, so we've got on the site of the old Radcliffe Infirmary, that penicillin was actually used in Oxford and in this former Radcliffe Infirmary to try and treat a patient with this antibiotic for the first time. And this patient, so the first person to be treated with penicillin, was actually a police officer in Oxford called Albert Alexander, who had an abscess in his mouth that turned into a wider systemic infection. And this started off looking really positive, so they treated him with penicillin and he started to get better. Great. But this was before there was kind of a good high throughput process for purifying penicillin, so they didn't actually have enough to fully treat this infection. So it started off really well, but there was maybe a kind of few milligrams total stock of penicillin in the world, and unfortunately this police officer died. But this wasn't a problem for much longer, and I think this is a neat or kind of a nice example of what's kind of happened with antibiotics. Is so the worldwide stock of penicillin in 1941 is estimated to have been around a few milligrams. But by 1945, around 4,000 kilograms of penicillin were being produced every month. And it was a really important drug that was used kind of on the front lines during World War II. And so the Nobel Prize in Physiology and Medicine went to Fleming, Chain, and Florey in 1945 for their work on penicillin. And I think what's really interesting is, so there's this quite famous quote from Fleming from a Nobel Prize lecture saying basically, it's not difficult to make microbes resistant to penicillin in the lab. And we've occasionally observed the same thing in patients. And it was kind of a forewarning on, you can kind of easily see that there's going to be a problem with resistance if we've observed this so far, depending on how we use antibiotics. And since penicillin, we've seen a bunch of other classes of antibiotics be discovered and come to the market and come to use. And kind of just as Fleming predicted, we're seeing this problem of antibiotic resistance on the rise. So I think this is really kind of a pressing global health threat, are the levels of antibiotic resistance. And there's been a lot of effort to put in to try and quantify the extent of this problem. And I think a really impactful paper came out 2 years ago in The Lancet doing exactly this. So to try and quantify and estimate what is the global burden of deaths that we're seeing that are already attributable or associated with antibiotic resistance. And from this paper, they estimated that already at least 1 million deaths a year are directly attributable to antibiotic resistance, and a further 4 million or so are associated with resistance. And this number at the moment is only predicted to rise. So I'm showing up here an estimate that deaths from antibiotic-resistant infections are estimated to climb to 10 million deaths a year by 2050, which, to kind of put it into perspective, would bring it into line with cancer. But how is this actually happening? So let's peel back a second, dig into the biology. So as a bit of an intro, so how do antibiotics work? They broadly work by killing or inhibiting the replication of bacteria, and they achieve this by a number of different mechanisms, but the common thread is they're generally targeting something that's pretty important for cell growth and cell function. So we see the beta-lactam antibiotics, so penicillin is a type of beta-lactam, targets cell wall synthesis. We've got antibiotics that target the machinery of protein synthesis in the cell. We've got those that target nucleic acids, so DNA synthesis. And we've got antibiotics that target the cell membrane. So all pretty important things for a bacterial cell to function. And then if we think about, well, how do bacteria become resistant? How can they avoid these mechanisms of killing? Broadly, we see a number of different mechanisms. So I'll give you a couple of examples, so kind of common mechanism of resistance might be the increased activity of efflux pumps. So bacteria have these pumps sitting on the cell membranes that can pump things out of the cells, and they can increase the activity of these, and so antibiotics may come in, but they'll get pumped straight back out. We might see, so the cell might be able to produce an enzyme that can inactivate the antibiotic and then it no longer works. We see target alterations, so mutations in the DNA and changes to the structures of the things that these antibiotics are targeting can provide a mechanism of escape. And we can also see decreased uptake. So for antibiotics that need to go into the cell to work, there's certain structures that let them into the bacterial cell, and so obviously if you're kind of reducing the uptake there, that's another common mechanism of resistance that we can see. And well, how do these different mechanisms of resistance emerge and then spread in bacterial populations? Two major mechanisms we think of here. We've got de novo resistance mutations, so spontaneous mutations. What I mean by this is, so DNA, whenever it replicates, just by chance, mutations, errors are introduced. And just by chance, some of these mutations may provide enhanced resistance to an antibiotic. So if then a cell or a population of cells that have this mutation are treated with an antibiotic, that's going to be a really strong selective pressure for those cells that can survive the treatment. And so then this mutation will spread throughout the population. And you can clearly see this kind of as a link for kind of target alterations, for example. There's also a process called horizontal gene transfer. So bacteria can spread and share DNA with their neighbors. And so you can imagine maybe there's a gene that provides enhanced resistance to an antibiotic. Maybe, for example, so we've got a kind of classic example of this is things called beta-lactamase enzymes that can inactivate beta-lactam antibiotics. And so these genes can spread between bacterial cells, for example, maybe on plasmids, and this can spread throughout a population. So that's a bit of a brief kind of overview and introduction to antibiotics, kind of with their history, mechanisms of action, and mechanisms of resistance. And now to introduce a bit of the theme that I work on. So our understanding of kind of how antibiotics work and a lot about how resistance to them evolves is historically and typically based on experiments where bacterial pathogens are exposed to antibiotics in isolation. So a classic example of this is something called experimental evolution, where you say, take a flask with media, you've got a single strain of your bacterial pathogen growing in this flask in isolation, and then you expose it to increasing concentrations of antibiotics, and then you look at the end point and you see, okay, how is the population that's there, how has it survived, what's happened, what's changed about it? And this is really fantastic, and this research has allowed us to build a really good and fundamental understanding of the mechanisms by which resistance can evolve, but doesn't fully capture the complexities of the biological environment of infection. And this is the area that my research is interested in, and I'm going to talk to you today about two examples from my research where we try and bridge this gap between these fundamental experiments in the lab and trying to understand what's happening clinically. And I'm going to tell you about two examples. So firstly, what can we learn from going in and studying resistance evolution within patients? And then how can we take this knowledge about thinking, well, what are the differences in these environments? And then apply that to experiments in the laboratory where we can try and dissect these variables in a controlled environment and learn more about the kind of processes behind this. Specifically, the example I'm going to tell you about is looking at the interface between antibiotic resistance and the interactions with other bugs in the microbiome. So, a second ago, I showed you this graph from this Lancet paper looking at estimating the deaths attributable to antibiotic resistance in this big global study. And if we look again in a bit more detail at this graph, what's going on this axis over here is it's showing deaths associated with infectious syndrome. And if we look furthest to the left-hand side of this graph, so kind of associated with the largest burdens, what we're seeing is lower respiratory infections. So infections in the lungs, respiratory infections, for example, like acute pneumonia infections, which are the type of infections that I'm particularly interested in. So we're seeing this big burden over here. And so this, I think, kind of highlights the important question of, well, trying to understand how does antibiotic resistance evolve during respiratory infections in this niche of the lungs. And so the first story I'm going to tell you about is a story from my postdoc here in Oxford in the Maclean lab in the biology department, where we're interested in exactly this question, and we're involved in this European consortium of a bunch of different hospitals across Europe. And the bug we were specifically interested in was the opportunistic bacterial pathogen Pseudomonas aeruginosa. So Pseudomonas aeruginosa is a major cause of hospital-acquired infections. So it's an opportunistic bug, so it's really problematic for people who are already quite sick. So we see it as a big problem in intensive care units. It's on the World Health Organization's critical priority list of pathogens in need of kind of critical need of new antibiotics, because it shows already very high levels of antibiotic resistance, and the infections are associated with significant mortality. And this picture here just shows an example. So these are some plates of Pseudomonas that I grew in the lab. So this is Pseudomonas samples that have come out of people's lungs. I think what's really interesting here is you can see this kind of big phenotypic diversity. So these different colors, they produce these different colored pigments and molecules called phenazines. And so we're already seeing kind of, they look really different when you grow them out on these plates. And the area we were specifically interested in was acute respiratory infections. So this differs to chronic infections like cystic fibrosis, and by acute I mean kind of short-term, fast-acting, the things that you see a lot of problems in intensive care units with. And so we were interested in ventilator-associated pneumonia. And what we were looking at, so we were looking at patients who come into the ICU, their lungs were getting sampled every couple of days, and we were looking at the Pseudomonas population within their lungs, and then we were piecing that together with the kind of patient history, which antibiotics have they been treated with, along with different measurements we were taking in the lab to try and dissect this picture and understand how this population of Pseudomonas is evolving antibiotic resistance in patient lungs. And the story I'm going to tell you about today is what happened when we looked across this whole patient cohort. So this ended up being a cohort of 35 patients, and we were looking at endotracheal aspirate, so this aspirate that's come out of their lungs. And we were looking at the population of Pseudomonas within their lungs, and specifically we were doing this by looking at 6 to 12 isolates of Pseudomonas per patient per time point. And what I mean by this, so what I mean, because I'm going to keep saying bacterial isolate. So when we're looking to kind of, if you imagine you've got a sample, imagine you've got this tube here, so you've got a sample and there's lots of different cell types, or there's lots of different cells in this sample, and you want to purify this sample and work out who's in there. What you'll do is you'll dilute this to a concentration where when you plate it out, you're seeing these distinct little circles. And these circles are called colonies, and this is a real kind of classical method in microbiology, is that we'll kind of dilute a sample down until we're seeing these colonies. And what these colonies are, it's when the sample is so dilute that a single cell has ended up on a position in a plate, and then it's grown to produce a pile of cells that there's so many cells now that it's visible to the human eye. And so then we say, okay, well, an isolate is what's grown from one bacterial cell on a plate. So it's kind of a distinct thing. And this illustrative example here, this plate shows a mixture of 3 different species. So for example, well, they're arrowed. We've got this one, and this one. And so you can see with this method, we're seeing them as distinct colonies. And so we were plating these samples out from patients' lungs, and for each plate we were interested in looking at 6 to 12 Pseudomonas isolates per patient sample. And this ended up with a total of 441 isolates. And this was kind of a really interesting thing about this project, because this was actually quite new to look at this population biology. So the classical thing in clinical microbiology is to look at 1 or sometimes 3 isolates per patient sample. And what we were doing with these, so we were measuring which antibiotics are they resistant to and to what extent. And we were doing this using minimum inhibitory concentration assays, where you basically plate your cells along here against an increasing antibiotic gradient, and you see kind of what is the minimum concentration of an antibiotic that inhibits the growth. This tells you about the resistance. And we were sequencing their genomes to work out, okay, what do they actually look like, who's there? And when we were sequencing these Pseudomonas genomes, specifically, we were interested in 2 things. So we were interested in the strain of Pseudomonas. So Pseudomonas is a species, and within a species, you're going to see a phylogeny of different strains. And we were looking at housekeeping genes, so really conserved genes within this species that all the members of the species will have, and we were looking for differences in these core genes to identify strains of Pseudomonas. And so these are things that are going to be really quite different to each other, so kind of thousands of SNPs and loads of different genes. So we're identifying different sequence types or strains of Pseudomonas within this population. And then we were looking at what changes over the course of the infection, so an analysis of mutations and any gene gain or loss during infections. And what did we expect going into this study? So the classical view of these sorts of acute respiratory infections is that these bacterial infections are going to be clonal, so a single strain. So the idea is that kind of a single entity or strain gets into the lungs, proliferates over the course of the infection, and so we expected that one patient would have one sequence type or strain of Pseudomonas. And if you think about this, this kind of feeds in as well with the classical microbiology view, where you'd classically only look at 1 to maybe 3 isolates per patient per time point. And we often expect in these scenarios that antibiotic resistance is going to emerge via selection for de novo mutation. So you've got this strain coming in, it comes into the lungs, it replicates, and just by chance, when it replicates, there is a mutation that, again, just by chance, provides enhanced resistance to a particular antibiotic. So if the patient is then treated with that antibiotic, it's going to be a really strong selective pressure for this lightning Pseudomonas that has this new mutation, and you're going to end up, so here, an infection with enhanced antibiotic resistance. So this is our classical view of these sorts of infections and resistance evolution in this environment. And we've seen good examples of where this happens in our single patient case studies so far. So we dug into 2 different patients who were really intensely sampled over a month, looked at kind of 100 different isolates of Pseudomonas in both of these cases, and it saw exactly this classical view. They had a single strain of Pseudomonas, and we saw resistance emerge via this classical de novo resistance evolution mechanism. But what happened when we looked across our patient cohort? So over here, I'm showing you a plot that shows patients along this axis and the number of isolates, so the Pseudomonas samples along here. And the colour of the bar represents the sequence type or strain of Pseudomonas. And so I think the kind of important thing when you look at this graph is you can see it splits into two halves. So you can see over here, all of these bars belonging to a single patient are a single colour. So we saw that the majority, so around 2/3 of patients in our patient cohort, were indeed colonised by a single strain of Pseudomonas, so our single strain populations. But we saw a surprisingly high frequency of what we've called mixed strain infections. So we saw around 1/3 of the patients in this cohort had not one, but 2 or 3 different strains of Pseudomonas in their lungs at the same time. And so, of course, what we wanted to ask with this is what is the impact of this within patient Pseudomonas diversity on antibiotic resistance evolution? And to do this, we looked at a subset of our patients, so patients that had been sampled on multiple days, so we could compare time points, and patients who had been treated with antibiotics that are supposed to be active against Pseudomonas infections. So this gave us a subset of 13 patients. And when we analysed these patients, we looked at classifying these isolates as either multi-drug resistant, so resistant to 3 or more antibiotics, or non-multi-drug resistant. And conveniently, this subset of 13 patients split into 6 patients with these mixed strain infections and 7 patients with single strain infections. And it's very nice when things happen like this, because this gave us kind of 2 almost equally sized pools to compare between. And our hypothesis going into this about what we expected is these mixed strain populations, purely as an artifact of being composed of different strains of Pseudomonas, are going to have an increased source of standing genetic variation. And we know from evolutionary biology that standing genetic variation is really important for evolution. So we thought in these populations that have these increased sources of standing genetic variation because they're composed of multiple strains, our hypothesis was that we may be able to see resistance emerging more rapidly in these patients. And so to look at this, what we did is for each patient, so we've got mixed strain in orange, single strain in blue, we looked at the initial prevalence of resistance isolates, so resistant isolates at the first time point, and what was the change in the prevalence of the resistance isolates over the course of this infection being treated. And kind of what you can see nicely from this graph is that within a patient, when initial resistance is low, we're seeing larger changes, larger increases in resistance, which makes sense, so that's nice. But importantly, when we corrected for this effect of initial resistance, and this was kind of our key point, is that we saw that these mixed strain populations were associated with significantly larger increases in antibiotic resistance. So kind of for any given resistance, initial resistance level, we're seeing a kind of 20% larger increase in resistance within these mixed strain populations compared to their single strain counterparts. So this was our key finding. Mixed strain populations are associated with the accelerated emergence of resistance. But of course, why? How is this happening? What are the drivers of this process? So to look at this, I'm just showing you an example of how we kind of analysed this patient data. So for each patient, this plot here is showing the time between the first and last sample. And at the first sample, what was the percentage of multi-drug resistant isolates in their lungs versus at the last sample, what was the percentage of multi-drug resistant isolates in their lungs. And this bar here, the colour of the blocks here, indicate the different antibiotics that they were treated with. And so I think what's quite striking from here is you can see this patient, we've only got 16 days but you can see they're treated with an incredibly large diversity and amount of antibiotics during these 16 days. And we're seeing the percentage of multi-drug resistant isolates increasing from around 65% to 100% at the end of the treatment. And why is this happening? So if we look at these inset pies, this is telling you which different strains of Pseudomonas were there. And we're seeing at day 0, there's 2 different strains of Pseudomonas in their lungs, and one of them is multi-drug resistant. So this is saying kind of 100% of these isolates were multi-drug resistant. And then the other one, this purple one, none of the purple isolates were multi-drug resistant. This patient is treated with all the antibiotics. Then what you see on this last time point is that 100% of the isolates are now this high-risk multi-drug resistant lineage. So this is an example of when what's driving these changes in resistance is changes in strain composition. So it's selection on pre-existing diversity in their lungs in terms of who is there Pseudomonas-wise. And we were able to kind of partition these changes into resistance into either these changes in strain composition, like the example I've just shown, or within strain changes, so our classical mechanism of de novo resistance evolution. So we could see in these mixed strain populations, when is resistance increasing due to within strain changes like mutations, versus when is it changing due to selection on strain composition. And we saw really predominantly that pretty much all of the time it's changing due to selection for pre-existing resistant strains. And that was kind of our real key finding there. And so to summarize all of that up, I think the kind of key take-homes from this study where we saw that mixed strain infections of Pseudomonas are surprisingly common in ICU patients. And this within-host strain diversity is really important because it impacts antibiotic resistance evolution significantly. And what we saw is that resistance is emerging rapidly in these patients that are colonized by mixed strain populations, and this is due to selection for pre-existing resistant strains. And I think the interesting thing here is this is when it was really kind of impactful to look at this population of Pseudomonas in patients' lungs. It was really important to look at kind of 6 to 12 isolates per patient per time point, otherwise we might have missed these dynamics. And I think a kind of interesting question this raises in terms of application is, could we measure Pseudomonas population diversity to help us predict the likelihood of antibiotic treatment failure? We still have quite a few questions about this project. So thinking about, well, how are patients getting mixed strain infections? When there's different strains of Pseudomonas in the lungs, where are they? And how are different strains interacting with each other? So those are just kind of some of the ideas that came off it. But I'm going to change gears now, and I'm going to talk about the second example from my research. So that kind of laid the groundwork for my interest in antibiotics, antibiotic resistance, and then thinking about what's going on in a patient that's different to when we're measuring just with a pathogen by itself in a tube, and how can we kind of bridge this gap. And if we think about bacterial infections, so bacterial pathogens rarely exist in isolation. They're embedded in dynamic and broader communities of microbes, i.e. the microbiome. And so species interactions with other things around them are a fundamental component of bacterial infections. And this is really kind of the focus and interest of my research moving forwards. And this project was funded, this was kind of the work I started to do as part of this Calleva Centre research project. And so what I was interested in here is both sides of these coins. So thinking about the interface between antibiotic resistance and interactions in the microbiome, So how does antibiotic resistance shape the interactions between species? And conversely, how do species interactions then shape the evolution of antibiotic resistance? So we're taking these components of like real live infections and thinking about, well, what's happening in a patient when they have an infection, and then trying to dissect these mechanisms in a more controlled system in the lab. And so the first question we wanted to ask with this is, when and how does antibiotic resistance alter the ability of a bacterial pathogen to invade the microbiome? And if we think back to the start of this talk, so historically, many antibiotics were originally isolated from microbes or modifications of microbial products. So obviously we saw that with penicillin from the penicillium fungi. We've also got the antibiotic streptomycin from Streptomyces and many other examples. And these compounds, they mediate bacterial competition in their natural capacity. So they're often thought to be kind of nasty things that bugs do to each other to gain an advantage in that environment. So if you think about that, a first simple hypothesis we can make about this question is that antibiotic resistance may also make you less susceptible to the things that your competitors do. But it's not quite that simple. So I've given you an example there of kind of a cross-resistance expectation, so where antibiotic resistance may also correlate to you being more resistant to the potentially nasty things that your microbial competitors are doing to you. And in this case, we might see antibiotic resistance correlate with an increased invasion ability. You can also imagine there might be scenarios of co-resistance where genes that encode antibiotic resistance are carried on mobile genetic elements that also have genes that provide a selective advantage in invading the microbiome. But on the other hand, antibiotic resistance, we know from a lot of studies, it's often associated with a fitness cost, and in this scenario you might expect a reduced invasion ability. And we also know about mechanisms of collateral sensitivity, so when you're more resistant to an antibiotic but more sensitive to a different antibiotic or another compound. And how all these factors trade off against each other is unknown, but clearly has implications for thinking about the ecological consequences of antibiotic resistance and the ability of resistant pathogens to succeed and spread onwards. So this is the question that I was really interested in, and as a model system for this work, we were using, kind of building on the previous project, Pseudomonas aeruginosa, in the context of the respiratory microbiome. So how does antibiotic resistance alter the ability of Pseudomonas to invade the microbiome was our question. And I'll highlight here two students. So this project started off with Lena Linden in her master's project, and then continued onwards with Sarah Shah, who was a research intern on the Calleva project with me over the summer, and did a fantastic amount to move this project forwards over the summer. And so what we were doing in this project is we were generating spontaneous antibiotic-resistant mutants of Pseudomonas. So we were taking a GFP-tagged strain of Pseudomonas so we could measure it when it was in a culture with other bugs, because it's got this GFP tag that the other bugs don't have. And Lena selected antibiotic-resistant mutants of this strain on 3 important antibiotics for treating Pseudomonas infections in the clinic. So she selected a total of 9 resistant mutants, and then what we did with these is we measured, okay, how resistant are they to these different antibiotics, so to validate this antibiotic resistance. Genome sequencing, how has this resistance happened? And invasion assays, where we were measuring the ability of these resistant strains to invade cultures of different bacteria from the respiratory microbiome that we could then compare to this ancestral wild-type background. And so this invasion assay, what we were doing is we were putting these different respiratory microbes in big 96-well plates, so we could do this high throughput and kind of do 96 invasions at the same time. We were allowing the respiratory microbe to grow in this environment for 24 hours, and then we were inoculating our GFP-tagged Pseudomonas strains in. So we were doing this for all the different resistance mutants, but also the wild-type backgrounds, allowing them to grow for 24 hours and seeing how well could they invade, compared to growth in just a rich lab media as a control. And to briefly take you through the results about what we've seen there, is we've seen examples of where antibiotic resistance is indeed associated with reduced invasion ability into these respiratory microbes. So we've seen for two different bugs, Rothia and Staphylococcus epidermidis, that the Pseudomonas resistant mutants to ciprofloxacin had a reduced ability to invade into these cultures. And these ciprofloxacin-resistant mutants had mutations in a transcriptional regulator that represses a multi-drug efflux pump. On the other hand, we saw a very clear example of where antibiotic resistance in Pseudomonas was actually associated with an improved invasion ability into a respiratory microbe, and this was seen with the bug Staphylococcus lugdunensis. So we saw here that the kind of normal background Pseudomonas strain couldn't actually grow at all in this culture in 24 hours, but the resistant mutants could. We increased this assay time because it looked like maybe we're just missing these bugs starting to creep in to 36 hours. Now, the kind of wild-type background could invade, but again, these resistant mutants were doing much better in this environment, and these mutants, again, had a combination of different mutations that would lead to increased multi-drug efflux pump activity. So what happens when we put this together? We've seen these Pseudomonas ciprofloxacin-resistant mutants that have this increased multi-drug efflux pump activity. We've seen they do worse in some respiratory microbes, but we've seen they do better in other respiratory microbes. So worse in Rothia, but better in Staphylococcus lugdunensis. So why do we think this is? So we dug into the literature a bit, and we saw, so for Rothia, we found a very interesting paper that said actually Pseudomonas is able to utilize metabolites that this Rothia bug secretes. And so Pseudomonas can use these Rothia-produced metabolites as a growth source and facilitate growth via cross-feeding. On the other hand, we thought this phenotype here, when we're seeing the improved invasion and the kind of inhibition of Pseudomonas in this environment, we thought maybe this could point to Staphylococcus lugdunensis secreting an inhibitory molecule into the environment around it, which is inhibiting the growth of Pseudomonas. So we put this together to a leading hypothesis, which says that our idea here is, so we're getting these mutations in this NFXB. This is leading to increased multi-drug efflux pump activity. These efflux pumps can efflux antibiotics, and that's why they're more resistant to ciprofloxacin, but can also efflux these Rothia-produced metabolites, explaining the reduced invasion ability into Rothia, as well as these potential Staphylococcus lugdunensis inhibitory molecules, perhaps explaining why there's improved invasion ability in the presence of Staphylococcus. And our next steps moving forward are to test this mechanism. Then finally, I thought I'd also highlight, we found examples of Streptococcus species. So these are bugs that are really commonly found in your oral microbiome, but also in your lower respiratory tract microbiome, that were able to provide resistance to Pseudomonas, and this was regardless of the resistance genotype, and this was observed at both 24 hours and 36 hours after inoculation. So to wrap all of that up, what are the key take-homes from this? So in this project, we saw that yes, indeed, antibiotic resistance can alter the ability of Pseudomonas to invade respiratory microbiome members. And we see this in both positive and negative directions, which are both Pseudomonas genotype and respiratory microbe identity dependent. We think that multi-drug efflux pumps might be important at this interface, and we're digging into this a bit more now. And thinking about, well, why do we care about these quite specific microbiome interactions, maybe? So the idea here is that could we use this to identify high-risk mutations that are associated with both increased resistance, but also improved fitness in the microbiome? Can we find signatures of a microbiome that might be more susceptible to resistant strain invasion? And can we identify microbes that can provide resistance or inhibition to both resistant and sensitive pathogen strains, which is obviously important for thinking about ideas of microbiome manipulation. So I'm going to finally wrap up briefly for the last 5 minutes or so with some perspective across the field. So what's going on in this space of antibiotic resistance for thinking about how can we combat antibiotic resistance moving forwards. So, and this is the kind of ideas that I'm interested in. So firstly, well, if we understand how antibiotic resistance evolves during infections, can we stop it? Can we, by improving our knowledge of how antibiotic resistance evolves, can we apply that to predict and ultimately control the evolution of resistance? As an example of an idea connecting the spaces there. So in a previous project, something we've looked at with these Pseudomonas infections is we were looking at the guts and the lungs of patients. And for example, we saw that gut carriage of Pseudomonas, so Pseudomonas could evolve, increased resistance in the gut, and then migrate to the lungs, and you could end up with an antibiotic-resistant lung infection. And so an idea off that is, well, if we know this is happening in the gut, can we target gut colonization before it can move up to the lungs? Another field where there's a lot of really cool research at the moment is bacteriophage or phage therapy. So just like we can be infected by viruses, and our cells can be infected by viruses, bacterial cells can as well. So bacteriophages are the viruses that can infect bacterial cells, and similarly, it's they get in, take over the replication machinery, loads and loads of more phage are produced, and then lyse and burst the cell open. So there's a lot of cool research going on here about can we use customized bacteriophage cocktails to target antibiotic-resistant infections in patients. Again, another field that I'm interested in, so this idea of microbiome manipulation, touched on those topics with the work I've just presented, but the broad idea here, and there's lots of really exciting research in this field, is can we use bugs to fight other bugs? So a classic famous example of this, of success, is using fecal microbiome transplantation to treat these sort of nasty chronic Clostridium difficile infections. So there's been success stories with patients here by transplanting the healthy gut microbiome from a patient or a healthy person without this infection into someone who has this chronic reoccurring Clostridium infection, has been successful in getting rid of this chronic pathogenic infection. Yeah, so I've popped a paper here which has, I thought, quite a funny name. So I think it would be a miss, again, if I was to give a talk in Magdalen and not talk about vaccines. So obviously there's heaps of cool research looking at can we develop vaccines against the big problem, antibiotic-resistant bacterial pathogens, with the idea that obviously if someone is vaccinated against, say, Pseudomonas, this is going to massively reduce the need for antibiotics. And so we're lucky here in Oxford, so there's the Jenner Institute and obviously also the Oxford Vaccine Group, and there's 2 Magdalen Fellows who are really world leaders in this vaccine development space. So Adrian Hill and Maheshi Ramasamy. Then the final idea I'm going to touch on, and you've probably seen posters about this whenever you visit, say, your GP, is antibiotic stewardship and infection control. So antibiotic stewardship being with the aim of reducing the unnecessary consumption of antibiotics, and thinking about core elements here. So kind of, okay, prescribing antibiotics when they're truly needed, prescribing them for bacterial infections and not, say, viral infections, making sure the appropriate and adequate doses are prescribed, and, well, yes, and the appropriate antibiotics for that bug are used. And infection control as well is obviously a huge element of this. And I think a really nice kind of potentially good news paper that came out a couple of months ago from this big nationwide survey in Spain was basically saying in 2017 the picture was pretty bleak, and this is, so the dark red is showing the distribution of extremely drug-resistant Pseudomonas across Spain. So we're seeing lots of the dark red over here. We fast forward 5 years, and the kind of nationwide antibiotic resistance policies seem to have potentially done something good here, and we're seeing a kind of, on a national level, a reduction in extremely drug-resistant Pseudomonas, which I think is a really cool paper to come out over the last couple of months that shows potentially some good news there. So I'm going to wrap things up and say, firstly, thank you very much for your attention and coming to the talk. If you're interested in reading more about the 2 stories I presented today, there's a paper and a preprint that we're in the process of taking through revisions. Acknowledgement and thanks to all the co-authors, colleagues and collaborators that are across this slide. Particularly with the last project that I presented, a huge amount of the work was done by these 2 students. So Lena Linden, who is a master's student, and Sarah Shah, who was a Calleva-funded research intern with me over the summer. A really big thank you to the Calleva Centre for funding this research. It's been really impactful and beneficial for me, especially as an early career researcher, to be able to get these opportunities to pursue my kind of own ideas and own research directions, and that's been really fantastic. I think, as mentioned at the start of the talk, I've spent the last few years as a junior research fellow here in Magdalen, and a couple of weeks ago I moved to Queen's University Belfast for a faculty job there and to set up my lab. So if you're interested in PhD opportunities or any of the work presented today, please feel free to get in touch. And a huge thank you for listening, and I'm happy to take questions.
Thank you so much, Rachel. That was wonderful. Yes, and thank you for taking questions. Other questions?
Thank you. Thank you so much for the talk. That was just amazing. I was wondering, so when we're talking about new approaches like phage therapies or using bugs to fight the bugs, is there any potential for development of phage resistance or bug resistance? For example, the phage invades the pathogen through the secretion systems. Like the secretion systems could be mutated or modified. How rarely or how often does this resistance appear or emerge? And is it any better than the antibiotic resistance?
That's a brilliant question. Yeah, so you're exactly right. Phage therapy looks really great, but obviously similarly to antibiotics, anything that's kind of killing or inhibiting the growth of bacteria is going to provide a strong selective pressure for escape. So that's something you definitely see, like you can observe it really easily in the lab. and it's a really like active field of research. We're only, I think, really beginning to chip the iceberg on the huge consortia of phage-resistant mechanisms there are. Like you've heard of CRISPR-Cas? That's a classic one, genetic engineering, but used in bacteria as like a natural immune system to defend against phage. So we're really beginning to learn about these different mechanisms. I think it's a really growing field, so I think it's only really in the last couple of years we're starting to see some really good like meta-analyses and reviews coming out, because there's lots of different centres around the world that are starting to use and trial phage therapy. So there's a really big one in Belgium, for example, and there's been some really neat papers recently like looking at kind of reviewing, well, what's the like collective evidence that we have for this working and how successful is this being in patients? And we're starting to see that yes, it is being successful in patients, not all the time. And when it's not always clear, right? It depends what they're looking at in that system. So I think there's exactly the same kind of risks of bacterial pathogens becoming phage resistant, but I think it, particularly for chronic or already antibiotic resistant infections, I think it's potentially a really good solution there, where you've just got to think, well, what can we do?
Hi, I'm a PhD student and I don't work on bacteria, I work on viruses. So I really liked your talk, especially the study on the patients. And so I was really curious about like the appearance of mixed strain pathogens within patients. And you know how in like viruses, whenever they get transmitted, there's like a tight bottleneck transmission that's happening, such that like one or two virions having different genomes, they are transmitted from one person to another. So I was just curious whether like these bottleneck transmissions, do they also occur in bacteria as well? Have they been studied, or that could be one of the reasons for the appearance of mixed strains within patients? So
yeah, great point. So I think that's exactly in terms of with that like classical viewpoint I put up at the front. That's exactly why we think, or why the kind of classical, well, there is a big bottleneck during transmission. So there's this big bottleneck of transmission, and I think that's a real kind of causative factor of why we do see a lot of clonal infections. And then you were saying, could the bottleneck be related to mixed strains or other strains getting in there?
So like in viruses, like there are just one or two genomes that are getting transmitted and then they expand in the recipient. So I was just wondering like whether this happens in bacteria as well. Maybe two strains, they start to like increase the number within the recipient population.
Yeah, for sure. I think we don't really know. I think it's a bit of an open. I think it's trying to capture, or like trying to even, with the caveat, I don't know much about the virus space, but I'm just thinking about like how we look at these things in patients, and it's like quite often you're sampling after somebody's quite sick, and it's quite hard. You don't know when you're picking up, or like when you start sampling something in the ICU, you don't know, has that just come in? Has that come in from outside? It's hard to capture those initial stages, but I think it definitely could be that maybe two things are coming in at the same time. I think something that we think is like high infection rate settings are probably quite important, in that like if you've got a setting where, well, there's a high infection rate, so there's lots of Pseudomonas potentially flying around and coming in. That could be behind mixed strain infections. Yeah, so I think it's an interesting space.
Hi, excellent talk. Thanks for giving it. I remember reading something in passing a few years ago about going back to the idea of like phage resistance and resistance to alternative treatments. I think I read something about how there's a sort of evolutionary tension between resistance to antibiotics and resistance to bacteriophages, where if a pathogen has a high resistance to one, they're more likely to be susceptible to the other. Do you have any idea how accurate that is?
Do you know what? I wish I did. I feel, yes, I'm not super up to date on the phage resistance space, but that sounds interesting.
Very good talk. So I have 3 questions for your first project. First is in terms of study design. So was there any specific reason to culture the respiratory specimen first and then sequence the isolates, other than just sequence the specimen right away? And the second one is I think to sequence the mixed strain, you used whole genome sequencing. Would nanopore targeted sequencing be any better in terms of a 2-hour turnaround time to expand because your cohort only included 35 patients, and in order to expand the study to a larger scale, would nanopore sequencing be more applicable in terms of mixed strain sequencing? And the third question is...
Oh, sorry, I'll answer the first one first, because then I'll just I'll forget the order. So the first question was, why did we do isolates rather than like whole sample sequencing? So the reason there was so we could link phenotype to genotype nicely. It's so we could look at 6 to 12, and for those isolates, we could say, okay, we've measured antibiotic resistance, and then we can link that with a genome. I think moving forward, it would be really neat to do a combination, right? To do sequencing of the whole sample and then the isolates. I think, well, at least when this study was set up, part of the study protocol was we were getting sent isolates rather than biological samples, because I think there's different kind of rules and regulations about how those sort of samples are moved between different sites. Does that answer your question?
Yes, it did. And the second one is you used whole genome sequencing for mixed strain. Would it be any better to use nanopore targeted sequencing for the strain like you're interested in, which gives about 10 hour time, 2 hours? So that might be more applicable if you want to expand the studies to multiple sites, maybe including more than with more patients instead of 35.
Yes, I think that's a really nice point. So the work I presented there, so that study was part of my postdoc in the McLean lab here, and Craig, the PI of that project, has recently been awarded an ERC Advanced grant to expand it. So we were only able to kind of time and cost, as you say, we looked at a 35 patient cohort. There are isolate samples from a lot more patients in the freezer, and I guess Adam sat behind you is the postdoc moving forwards that research. So it's interesting to hear your thoughts there. And I think while we were towards the end of the project, we were thinking about what would we do moving forwards and kind of what's the mix of sample sequencing versus kind of what's the most efficient way to work out who's in a sample and also do that high throughput phenotyping at the same time.
Okay, so and the third question would be how positive or how soon you think your pipeline of surveillance of this Pseudomonas developing, because Pseudomonas is killer for ventilated patients because it's got less wellness in terms of escalating antibiotics compared to Klebsiella. So how positive your pipeline would be, the whole surveillance of that would be implanted in a clinical trial to at least to see how it works in terms of alter, how quickly the decision making. No, how would that affect the decision making from clinicians and also eventually the mortality reduction?
So is your question there thinking about, so we've seen mixed strain, we've seen this link with mixed strain populations and accelerated resistance emergence, kind of what are we going to do about that moving forwards?
Yes.
Yes, okay. So I think that is going to be a big focus of this ERC Advanced grant, is thinking about, so we saw this in this, well, it's going to be a focus of that, so we saw this in this cohort of 35 patients, but realistically when we kind of had to narrow that down to a subset of 13 patients where we had the start point and the end point, and in that cohort the results we got were very significant, but what happens when we look across the rest of the population? So we'll be looking into that there. I think in terms of ideas of like how you would bring that forward, I think kind of linking to your second question, thinking about like what's the most high throughput and efficient way to capture the diversity status of Pseudomonas in the lungs is a big question, and thinking about the genetics. So for example, we looked at it with this multi-locus sequence typing where you're looking at 7 housekeeping genes. Is there a simpler way to do that? Can we find a new, maybe like one region of the genome that says you sequence it and you say, okay, that's who's there? But I think these are all interesting questions moving forward, so thank you.
Hi, Rachel, nice talk. I have just one very quick question. So when you were trying to look at how well the pathogen invades an established symbiont, what you were doing was to invade the pathogen inside media that has already been grown with the symbionts. So presumably that would mean that the media would be nutrient depleted. So the first question I have is, are these nutrient depleted conditions representative of what you would expect in the lungs, for example?
So this is for the second project.
Yep.
Yes, so you're right. What we were doing is we were growing up a microbiome strain in media in the lab, allowing it to grow for 24 hours and then inoculating Pseudomonas in. How representative is it of the lungs? I think it depends kind of what sort of, I think there's a huge amount of variables there, right? I think the lung microbiome is lower density than places like the gut. I think we don't have, there's being some good kind of respiratory mimicking medias being developed now, but we don't really have a great idea all the time of when things are in, compared to the gut at least, like when things are in the lungs, like what are they eating? What are the nutrients there? How do the presence of other things shape that? So I think, well, at least the idea behind the assay was to be able to develop kind of a screening assay where we could link specific resistance genotypes and phenotypes with specific invasion phenotypes off the back of that, and just ask, does resistance play a role in shaping the outcomes of these species interactions? Moving the forwards, and I'm thinking about doing this now as I'm setting up the lab in Belfast, I'm thinking about like, what is the best way to take this project forwards? Thinking about using in vivo host infection models and exploring the space there.
Yeah, I think that makes sense. I was also wondering of why you wouldn't have them, for example, grow at the same time, so they would then reflect competitions where they are fighting for the nutrients. Because presumably if the pathogens are also handicapped in a way, with the plasmid or whatever resistance they are carrying, then maybe their ability to acquire these nutrients would be impacted as well.
So the reason we grew up the microbiome strain first was to think about this idea of microbiome invasion, right? To think about a scenario where you've got the microbiome that's already there, then you've got a pathogen coming in. When we inoculated the Pseudomonas in, they were growing together then for an extra 24 hours. But it was to think about this idea of invasion. But indeed, kind of another interesting question is you can ask all of these sort of variables, right? You can think about what does it look like when they invade? What does it look like when they're growing at the same time? What does it look like when it's the other way around and it's maybe, say, you've got a fully established Pseudomonas infection and then maybe you've got different microbiome members coming back in.
Yeah, thank you.
Oh yeah, I can answer. Hi Rachel, thank you so much, that was really good. I was wondering, so when you talk about microbiome interactions as a potential therapy or preventative for infections, in terms of the lung microbiome, what would this look like? Because I guess we know that you can give probiotics for the gut, but in terms of the lung, would it be a nasal spray, or how would it look?
Yeah, so there's some cool pilot trials of this out there if you dig into the literature, and it's looking at aerosolized things, as you say, like nasal spray or aerosolized. And we know that for the respiratory microbiome, so you've got your upper respiratory tract and your lower respiratory tract, and what's going on in your lower respiratory tract, it looks very similar to what's going on in your upper at a lower density. It looks very similar to the oropharynx microbiome space in particular. So putting things kind of doing well in your oral or nasal microbiome kind of feeds down.
Thank you.
Can I ask a final question to finish off? So, in terms of the ways in which mixed strain infections or the presence of mixed strains drives enhanced development of antibiotic resistance, is there any data yet available on what types of mechanisms evolve more quickly? Do you see what I mean? So whether it's particular, are there particular kinds of mechanisms of resistance that seem to be more adept at evolving more rapidly, or maybe maybe it involves mixtures of types of. Do you see what I mean? It's just because as a biochemist, it's sort of interesting to think about mechanism and what it might be about different kinds of mechanisms, whether it's efflux pumps or metabolizing enzymes or what, that might evolve more rapidly or might be more adaptable to enable a bacterium to acquire a novel kind of resistance, and whether some evolve more rapidly as a result because they're basic mechanisms, or I don't know whether.
No, good question. So that is a very interesting question. With the patient data we've got at the moment, there wasn't enough resolution to dig into that. So I think basically what we saw the kind of main significant factor that came out of the analysis was that just having mixed multiple strains there, resistance is emerging more rapidly with our phenotypic measures. When we narrowed that down, that was comparing 6 mixed strain infections to 7 single strain infections, and the mixed strain infections had these kind of multi-drug or extremely drug-resistant bugs in there that have a huge repertoire of different resistant mechanisms. So some of them are having 10 different acquired resistance genes. And so we had a list off that of the things that are doing, kind of driving those resistance changes in those infections. We've got, okay, we know what these are, we've got their genomes, and we can say, okay, we've put them through this resistome analysis where we can say, these are the different SNPs they've got, and we know kind of what those mechanisms should look like, and these are the different acquired resistance genes they have. But because it was such high numbers of both resistance SNPs and resistance genes, we couldn't dig into, is there one mechanism that's more important than the other, and we didn't have the volume of patient data to look at that kind of big picture statistically either.
Sure. But although your answer does suggest, I might be looking at it the wrong way around, it might not be so much particular kinds of mechanism that evolve more rapidly, but that actually it's to do with the ability to acquire many different types of mechanism in some underlying way, some underlying mechanism that might allow multiple types of resistance to emerge in a particular strain. That could possibly be, and there could be some very fundamental aspects of biology that, being more adept at DNA acquisition or something horizontally, that could drive that. So that's. Thank you very much indeed. Thank you.
Thank you.
And let's thank Rachel again for a brilliant talk. Thank you. And yes, if you do have questions or would like to talk more with Rachel afterwards, then please do join us outside for a drink.